Today we see study of microscope. So this is practical from cell biology. So aim of this practical is that to study the microscope, that is simple and compound microscope. So agenda of this practical is that microscopy, what is microscopy, then simple microscope and compound microscope we see. Now let us first we see microscopy. What is microscopy? Micro means small and scope means to study. So, an instrument designed for visual examination of small object which cannot be examined by naked eye. So, that instrument we simply call it as a microscope, and the different types of such as the simple microscope, compound microscope, phase contrast, electron microscope that we are see in our day-to-day life. So the study of different microscope is called as microscopy. Now let us begins with simple microscope. So the simple microscope, it is commonly used in laboratory. It is nothing but magnifying glass. These microscope used when very small animal have to be dissected or for magnification of parts in the sections or of small organisms which are not seen by naked eye. So these microscopes simply we see the magnification parts of that section or the magnification part of that organism. So this simple microscope is made up of two parts, that is optical part which contains the convex lens and the mechanical part which holds the lens in proper position. Then the knob is for proper magnification. So see this picture. uh, then mirror for light passing, then clips for holding the slides. So this microscope is working like as a, it consists of convex lens which magnifies the object and helps to produce magnified image. Magnification obtained by the formula, that is capital M is equal to 250 plus 1 by F. So here small f is called as the focal length of lens in centimeter. Then 250 is a distance of distinct vision. See this picture is of simple microscope. It contains handle, then eyepiece for the observation, then it contains clips for the, it contains clips for to holding the object. Then it also contains stage, it is for to holding the again object on which the slide can place or the whatever material that can hold in this stage. Then adjustment screw is there for the adjustment purpose. Then mirror is there for light passing or light passing from sunlight from the direct from sunlight to the that object. Then is this is the base for holding the all microscope. This is all about the simple microscope. Now we study the our second microscope is that compound microscope, or simply it is called as light microscope. Recently we it called it as a light microscope. These are two microscope, means compound microscope are of two types. These are mainly the two differ in source of light. This difference is depend on the source of light, which light they use. So one uses a mirror to converge sunlight, while other uses directly an illuminator, means directly use the light. So this is the difference in these two microscope, or simply the light microscope. What is it? What is it that compound microscope? The term compound refers to the microscope having more than one lens. Compound means more than one lens. The term light refers to the method by which light transmits the image to your eye. So, the microscope is the combination of two words, micro meaning small and scope meaning to view. So, let us see the parts and their functions of this compound microscope. Firstly, first part is that eyepiece and body tube. The eyepiece is the lens through which the viewer looks to see the specimen. It is usually contains 10x or 15x power lens. Then the body tube connects the eyepiece to the objective lenses. Then objective and stage clips. Objective lenses are the one of the most important part of the compound microscope. They are the closest to the specimen. A standard microscope has 3 to 4 objective lenses which range from 4x to 100x. The stage clips are metal clips that hold the slide in a place. So this is about the objective, function of objective, and the stage and clips. Now we see the arm and base. So the arm connects the body tube to the base of the microscope. Then the base supports the microscope and its rear illuminator. So this is about arm and the base functioning. Then the illuminator and stage. Illuminator is the light source of microscope. A compound light microscope uses a low voltage bulb as an illuminator. Then stage is the flat platform where the slide is placed. Then nosepiece and aperture. So nosepiece is a rotating turret that holds the objective lenses. The viewer spins the nosepiece to select different objective lenses. The aperture is the middle of the stage that allows light from the illuminator to reach the specimen. This is the function of the nosepiece and the aperture. See, each and every part is highlighted in the slide, and it naming. Then we see the condenser and diaphragm. The condenser gathers and focuses light from the illuminator onto the specimen being viewed. So the condenser gathers and focuses light from the illuminator onto the specimen being viewed. The diaphragm is a 5-holded disc placed under the stage. Each hole is of a different diameter. By turning it, you can vary the amount of light passing through the stage opening. So here on our right hand side, we see the picture of compound microscope containing eyepiece, body tube, nosepiece, arms, objective lenses, stage clips, diaphragm, coarse adjustment knob, fine adjustment knob, illuminator, and the last one is the base. This is all about the compound microscope. So let us see how does light travel through a microscope. Light is first emitted by the light source and it is directed by the condenser lens onto the specimen. The light from the specimen then passes through the objective lens. Further, light rays are passed to a projector lens which reverses their direction so that when the image reaches the eye, it will not appear upside down. Not all microscopes have a projector lens, so the viewer may be seeing a reverse image. In these cases, when the slide is moved, it will appear to be moving in opposite direction to the viewer. The light rays then travel to the ocular lens or eyepiece. This is often a 10x magnification lens, meaning it magnifies the magnified image an additional 10 times. The image is then projected into the eye. In this way, the magnification is observed or magnification is obtained. So, see, this picture is totally about the magnification 4x that converts into the 40x, then 100x and 400x. So these compound microscope have a lots of uses. So few uses here we see uses of compound light microscope. First is the compound microscope is a great use in pathology labs so as to identify the various diseases. In forensic laboratories, compound light microscopes are used to identify presence of minerals or metals in human cells so as to solve criminal cases. Forensic experts can also find out the origin of drug by viewing its component particles under a microscope. So also the microscope is observed in schools also. Students in schools and colleges are benefited by the use of microscope for conducting their academic experiments. It is also it is of immense help as the students can now see the bacteria and viruses which is otherwise invisible to the naked eye. Thereby they witness things which they have studied in their theory. Plant cells and also the microorganisms living on them can be observed under a microscope. So, a compound microscope has proved beneficial to biologists too. So, these are the various types of cells that are observed under a microscope: blood cells, human cell, plant cell, that all are observed under the microscope. Without microscope we can't see the cells. Thank you.